Under every wheat field, sunlight is being folded into quiet, edible starch. In rice grains and potato flesh, glucose gathers as long, pale chains. Some chains run straight, but many branch like winter trees underground. Those branches make amylopectin compact, sturdy, and beautifully hard to unpack. For living cells, this architecture stores energy without flooding the surrounding world. For cooks and makers, it can become a locked pantry of sweetness. Ordinary amylases nibble along the straight roads of starch with patience. But at each branch point, their progress slows and finally stops. Small forked leftovers remain, called limit dextrins by later carbohydrate chemists. The problem was not scarcity, but access to hidden sweetness inside grain. Nature had met this problem long before human factories needed answers. Germinating barley releases debranching helpers as the sleeping seed awakens. Soil bacteria gather around fallen grain and loosen its branching energy stores. In animal intestines, microbes make quiet tools for difficult carbohydrates every day. Each community discovered that one tiny bond controlled the whole maze. That bond, an alpha-1,6 link, sits at every branching point in starch. Break it cleanly, and tangled starch becomes ordinary chains again. Pullulanase would become one of nature's elegant openers for these branches. The first clues came not from factories but from malted grain beds. In 1833, Payen and Persoz studied malt extract in Paris laboratories. They named diastase, an unseen agent that could liquefy starch. Their work helped reveal enzymes before anyone knew their molecular shapes. Across the 19th century, brewers watched warm mash change into fermentable sugar. Chemists saw dextrins appear, stubborn fragments between starch and full sweetness. The branch itself remained invisible, awaiting gentler 20th-century chemical methods. Another clue grew as a glossy film on fruit, leaves, and bark. A dark yeast, then called Pullularia pullulans, secreted a strange extracellular glucan. In 1938, researchers described this gummy material, later known as pullulan. Pullulan was simple in one way and deeply revealing in another. It carried repeated alpha-1,6 joins between short glucose stretches like tiny bridges. To chemists, pullulan became a clean model of a difficult branching bond. If something could split pullulan, it might also debranch the starch harvest. In 1961, Bender and Karl Wallenfels reported that something finally could. From Aerobacter aerogenes, they found an enzyme that attacked pullulan directly. They gave it the name pullulanase after the substrate that had exposed it. The organism's name later changed, but the enzyme's identity quietly endured. Pullulanase did not chew randomly through every glucose connection it encountered. It sought the branch linkage and opened the fork with remarkable restraint. Suddenly, starch chemistry had a dedicated key for an old molecular lock. Laboratories soon tested related bacterial debranching enzymes against amylopectin and resistant dextrins. The results connected a rare microbial talent to ordinary crops and harvests. A discovery from a culture flask began moving toward starch refineries. Industrial starch makers were already using enzymes to replace harsher chemical routes. Pullulanase added a missing touch, freeing branches other enzymes left behind. Later bacterial sources offered enzymes more comfortable in warm, acidic starch streams. Among them, Bacillus acidopulolyticus became important for dependable industrial pullulanase. The story shifted from biological curiosity to dependable partner in starch conversion. At the microscopic scale, pullulanase resembles a hand around a forked thread. The starch chain settles into a groove on the enzyme surface. The branch point is held just long enough for water to enter. One precise cut separates the side chain from the waiting backbone. Nothing is burned, forced, or wasted in that small catalytic moment. The enzyme lets go, unchanged, ready to find another hidden branch. Freed chains become easier for other enzymes to finish gently and completely. In pullulan, the same action releases neat pieces of maltotriose. In amylopectin, it opens dense clusters into simpler paths for sugars. Its power lies less in strength than in careful selectivity. It recognizes where to act, and just as importantly, where not to act. In sweetener plants, pullulanase helps starch give up more fermentable sugars. It supports glucose and maltose syrups used throughout familiar everyday foods. In brewing, it can help create cleaner fermentations from grain starch. In baking, it helps shape crumb texture and slow starling naturally. In rice and noodle processing, it can fine-tune starch behavior. In biotechnology, its released sugars can feed fermentation for renewable materials. For analytical scientists, pullulanase reveals branching patterns inside unfamiliar native starches. Its quiet usefulness depends on matching enzyme, material, temperature, and purpose. Pullulanase reminds us that green technology often begins with patient listening. A microbe solves a small problem, and industry learns useful humility. Instead of breaking matter with force, enzymes invite it to unfold. In a future built from renewable carbon, such invitations may matter deeply. At Enzymes Bio, scientists and enzyme enthusiasts work to make this biology easy to reach. One platform, many enzymes, no tangled procurement for research labs and growing businesses. A green technology with a long future. Learn more at enzymes.bio.